Thank you, Thomas. Thank you, Tom Bever, also. And thank you all for being here. And we're going to dive into the brain, in fact, inside neurons, for the next two talks. The first is remote. The second will be live. And do we have Christoph Simon online as yet? One more minute. So Christoph Simon is a biophysicist from the University of Calgary in Canada, Alberta, and he's been known for a number of years for looking for and finding photons in the brain, and he has some very interesting ideas about photons and entanglement, and I'm getting the thumbs up sign. So Christoph, are you there? Let's see you and take it from there. He's been to several of our conferences and is a really good guy. And is he there? There he is. Christoph, can you hear me?
Yes, can you hear me?
Yes, we can. You just disappeared, but we see your slides. So
okay,
if you're ready to go.
Almost, yeah, thank you very much.
Christoph Simon.
Thank you very much, Stuart. Yeah, so thanks for giving me the opportunity to talk to you here, even though I couldn't make it in person, which I regret. It would be nice to be in California now. But anyway, I'm here, so I'm in Calgary. I'm a quantum scientist, but I'm also in our Brain Institute, as you can see here at the top. Sorry, so my topic is, I think, consistent with the topic of the conference or the workshop as a whole. So whether quantum entanglement could somehow be important in the brain. So let me just, those of you who are not super familiar with it, let me just remind you what entanglement is. So it is a unique quantum property that allows two or more, can be many more, particles to act in correlated ways, even when they are separated in space. We know for a fact that these quantum correlations can't be explained in classical terms. That is the essence of Bayes' theorem. And the sort of key feature that I want to highlight here is that an entangled quantum system should really be thought of as a coherent whole, even if it contains many parts. You know, that is something that doesn't exist classically. So why would one sort of want to invoke entanglement in the context of the brain and of neuroscience? There's maybe more than one reason, and here are my favorite two. So on the one hand, you know, if you have sort of an engineering mindset, you might be convinced by the first one, which is many people, including me, you know, work on quantum technology and specifically quantum information processing. The community has found over the last few decades that it can be advantageous, right, to use quantum properties like entanglement for information processing. So it doesn't seem crazy to think that you know, nature might have figured that out too, as it has many other things, right? So maybe it found a way to harness these quantum features in our brains. Then the second argument is a bit more maybe for philosophically inclined people, right? And that's the binding problem of consciousness. It's difficult to understand how our experience, which is somehow unified in some sense, right? How that arises from this like you know, connection of neurons and really molecules that we have in our heads. So very difficult to understand classically how this binding could happen. But for entangled systems, as I said on the previous slide, you know, that the whole really is more than the sum of its parts. It really is more in a well-defined mathematical sense. So maybe that's a reason to take it seriously, and a second reason to take this idea seriously. Now, I am a sort of a quantum engineer, I guess, in my day job, right? So we think a lot about how to implement quantum information processing in our labs, or you know, in artificial systems. And sort of based on that, one could sort of try to reverse engineer the brain. If nature has had to do it, what seem like the most promising ingredients that it might be using based on our experience with technology. And that has led me personally to these two. On the one hand, photons, like we know that photons are, so that sort of light in the visible and near infrared spectrum, let's say that's what we have found to be ideal for sending quantum information over any kind of macroscopic distance. There really is not a good alternative to that, at least that we have discovered so far. And then on the other hand, spins, electron spins, and maybe even more so nuclear spins, are very good for quantum to serve as quantum memories and quantum processors. And we know that they can do that even in condensed matter systems and even at room temperature in some cases. So there really are, in principle, physical degrees of freedom that are well suited for this task. Of course, then the question is how could that work concretely, right, in our brains? And that's what the rest of this talk will be about, in some sense, at least taking some initial steps towards that. So let's start with the photons and then come to the spins afterwards. So there are photons that are emitted by our brains, really by cells in general. They are often called biophotons. It is thought that, or it is known that they are somehow closely related to reactive oxygen species in the brain or in cells. So they are definitely there. You can see them. This is an image of a brain slice without any external illumination, any really just spontaneous emission of photons. That's what we call biophotons. So they're definitely there. The question is whether they are somehow used by biology, right? If they serve a purpose, could they be used for communication somehow? We know that there is potential photon detectors in the brain. There is really more than one possibility, but a prominent one are opsins, light-sensitive proteins, similar to the ones that we have in our eyes. And I'll come back to that in a moment. I am here interested in quantum communication primarily, but of course, even if we just discovered that there is an active classical photonic sort of communication channel in the brain, that would already be very interesting. So one idea that we proposed in this context now about 7 years ago, in this paper here at the bottom, is that axons, which everybody agrees are kind of like electrical cables in the brain, right? That they are actually also really quite well suited to serve as optical fibers. So electrical cables means we are talking about electromagnetic signals in the very low frequency range, right? But here I'm talking about optical fibers that conduct visible or near infrared light, and we are suggesting that axons can really serve a dual purpose, like conducting both kinds of signals. We did a lot of, and the key observation is that the refractive index of axons is higher than that of the surrounding medium, which is more or less water. And then if the axons are myelinated, then the refractive index of the myelin is even higher still. And that is really what you need for light guidance. So we did a lot of pretty detailed modeling, which I think I will skip for this audience. But basically, we convinced ourselves, and I think to some extent other people, that this is really a sort of a reasonable possibility. And there is now even some experimental evidence. So these were our own estimates here, that we concluded that transmission, like with a good sort of success probability per photon in the millimeter to centimeter range, really seemed reasonable for realistic axons, and that would I should say that there's not a lot of photons that are emitted by a single neuron at any given time, right? But there are still billions of them that are emitted per second if you look at a human brain, and that would be a lot of quantum information that could potentially be shared in this way. If one thinks about, what is actually the bandwidth of conscious experience, there's, of course, that's not an easy question to answer, but it's definitely less than that. I think everybody agrees. So there's enough of these photons, and they could conceivably go, far enough to really make a difference in the brain. So is there any experimental evidence for this? Yeah, so there is various, actually. So there's some work that was done before that we were aware of when we proposed this idea first. For example, people had shown that so-called Müller cells that we have in our retinas, and that are quite similar to so these are glia cells, so they are sort of materially quite similar to the myelin sheath, which is also made by glia cells. And they are light guides in our retina. This is something that these authors showed in 2007. But there's also more direct evidence now related to the brain. So, for example, here, this is a paper from Canada from 2020, where you can see a brain slice, and you can that's illuminated from below, and you can really see that the myelin sheaths are brighter, right? So one can sort of see here directly that the light seems to be guided in this sheath, right, that surrounds the axons. So that's encouraging, and these authors pointed that out as well. It would still be good. So I think this shows that the basic physical principle of light guidance is probably correct. The quantitative details are still need to be studied. Like we don't know, can it really go for a few millimeters or a few centimeters? Like what's the transmission for a real axon and so on? That hasn't really been done, and we are sort of working on trying to make that happen. Those kinds of experiments. Yeah. So here is an example. I talked about opsins before. So this is a paper where they showed that light really can have biologically meaningful impact on neurons, even deep in the brain. These were neurons in the hypothalamus of mice, and they showed that when they were sort of activated by violet light was shown onto these neurons, then they changed the thermogenesis, which is basically the balance between doing work and producing heat, which is important for cells, right? So that balance was changed in response to the light. So quite an interesting effect. Then, so we were encouraged, by these various recent results, experimental results, to speculate a bit more, to illustrate maybe how the photons could help the brain. So for sure, like what we are really after is this quantum level, quantum information processing. But here's an example how even in the classical context, the photons could be useful. So one of the puzzles when people think about learning in the brain is we have a very powerful sort of paradigm for how learning can work now in artificial intelligence, which is called backpropagation, the backpropagation algorithm. But that requires, as the name suggests, it requires that somehow, so you have a network, and there's sort of a forward flow of information in the network, but from inputs to outputs. But then the backpropagation algorithm requires that information can also propagate backward through the network in an efficient way. And that's kind of difficult to see how that can work in the brain. And there's a whole field of people, studying how backpropagation could be implemented in the brain. And this idea of photons as information carriers is a potential solution for that, because the photons don't care, so the electrical signals are in some sense directional in the brain. They can't easily go backwards along the axons, but the photons can. An optical fiber is really sort of a symmetric thing. And so here we developed that idea in this paper, that the photons might be the ones who are in charge of back propagating the error signals. But this is speculation at this point. Okay, so coming to the second part. So I said, photons might be important, and the other really promising quantum systems in my mind are spins. And the way I want to start that part is by talking about magnetic field effects. So I'm not expecting you to read all these lines. I'm trying to convey that there's a lot of observations of effects of weak magnetic fields in the microtesla to millitesla range on a wide range of biological phenomena, okay, like going, and many of them in the brain, not limited to the brain, but many of them in the brain. So magnetic fields do a lot of things to biology. Here's more, frequency dependent and intensity dependent and so on. So lots and lots, and we have this review paper here that came out last year in the Journal for Society Interface, where we begin by reviewing, the many observations that people have made in that context. And the point is that this is not so easy to understand, in fact, from a classical perspective, because these weak magnetic fields that we are talking about here are weak. So the associated energies are very small. They're much smaller than thermal energies at body temperature. So you really wouldn't think that even from a classical molecular mechanics sort of perspective, you wouldn't expect these magnetic fields to do much to the chemistry. But there is quantum mechanisms, and in particular this one, which is called the radical pair mechanism, which is a potential explanation for how that might work. So the idea is that so you can have radical pairs, those are pairs of molecules with single electron, unpaired electrons. Like usually in biological molecules, the electrons are somehow paired up. But sometimes there's individual electrons, and they can sometimes be, they can be created, for example, by one electron hopping from one molecule to another. Then you have a pair of electrons that are correlated in spin, but they are spatially separated, but they are entangled. They can be entangled. They correlated and in some cases entangled. So now we have and that entanglement can then so you have the electron spins, but then there's also nuclear spins around, and in the presence of those nuclear spins, the dynamics of the electron spins then is influenced by external magnetic fields, it turns out. So now so these correlated electron spins are kind of like magnetic field sensors, and they will behave, the chemical reactions that they can undergo are going to depend on the spin states, because very, very simply, you can imagine that if the electron spins are opposite, then they can sort of pair up again, but if they are not, then they can't, and like the external magnetic field will change the balance between them being correlated or uncorrelated. I'm oversimplifying, okay? It's really about their quantum states, but something like that. So the spin states are going to determine the reaction products, and the spin states are influenced by the magnetic fields. So this is a sort of well-established mechanism in spin chemistry. It has been studied a fair amount in the context of magnetic field sensing by birds, and in particular by these avian magnetoreception, that's called. And here, where the focus has been mostly on the so-called cryptochrome proteins. But there is a growing realization that this may be a more general thing in biology. So there is on the one hand, for example, the cryptochromes are also implicated in axon growth in response to transcranial magnetic stimulation. On the other hand, even in the magnetoreception context, it seems that it doesn't necessarily have to be the cryptochromes. In fact, this was a recent paper here in Nature, although this is an ongoing debate. So in any case, we are suggesting, I guess, that this phenomenon, this radical pair mechanism is actually widespread in biology, and we have developed a number of example studies to make that concrete and believable. So I won't go into all the details on that, but I'll just sort of briefly mention. So we had our first foray into this field was on anesthesia. So xenon anesthesia, we think that there's a radical pair mechanism underlying that, where the evidence is really an isotope effect. That the potency of xenon as an anesthetic depends on the isotope in a way that shows that it's the nuclear spin, and that is in some sense a smoking gun, or an indication that this radical pair mechanism may be at work, and we are making predictions, that basically the anesthetic potency should therefore also depend on the external magnetic field, which would be an interesting experiment to do. There's a similar isotope effect for lithium treatment of bipolar disorder, and again we predict that there should be magnetic field effects. There are magnetic field effects that are known on the circadian clock in fruit flies, and so we suggest that that could be due to a radical pair mechanism, actually even in cryptochrome, which is sort of the classic mainstream protein for these kinds of effects. We made some predictions on that. And also in microtubules, which I know are dear to Stuart's heart, there is actually reports of magnetic field effects basically on microtubule assembly in vitro, which to our mind, again, suggests that there's radical pairs, and that's something that we are pursuing together with Travis Craddock, who I think is going to speak later in this meeting. So really many examples, right? And that's even just scratching the surface. So here's another paper on neurogenesis, where people saw that shielding the magnetic field, really influences neurogenesis through somehow reactive oxygen species, and we made a model for that. So yeah, so we wrote a number of papers on this, but they are summarized. Yeah, I'll show that at the end. So we have a review paper and sort of a manifesto about that, which I'll highlight again at the end. And we now have a number of experimental collaborations on this. So we are looking at biophotons and magnetic and delayed luminescence, which is sort of light not without any external illumination, but a long time after an external illumination. That's called delayed luminescence. So it's also very weak. We are looking at, for example, the magnetic field dependence of that, and we are finding interesting things. We have a collaboration with Wendy Bean in Michigan on magnetic field effects on stem cells in flatworms. Here, a collaboration in Calgary on pigmentation in tadpoles. So here's the microtubule project that I mentioned with Travis. So there's really a whole range of phenomena where if one looks, it seems relatively easily one finds these effects of magnetic fields, which we think are due to a quantum effect, really, right? This radical pair mechanism. So that's very interesting and encouraging. Here's one other thing. So coming back now to the maybe main theme of this meeting, and so the question of the brain and consciousness, like how what does that have? So maybe there's quantum phenomena at the cellular level, okay, but like how would the brain use it? Like why would that be helpful in any way, to harness that? Here's an example that could maybe be a hint, how it might work. So this is people are now interested in neuromorphic quantum computing. There's a field of people who are looking at that, and in particular, there's a paradigm called neuromorphic computing to begin with, right? Like so, and within neuromorphic computing, there's a paradigm called reservoir computing, and then now people are wondering, looking for signs of quantum advantage in these neuromorphic computing schemes, and here, so we are entering this field because it's relevant, right? So we found some evidence that if you have more quantumness in some sense in your reservoir, then it performs better. So that could be a hint of how quantumness helps the brain, learn better or various things like that. But this is new and unpublished for now. So yeah, but we do see a correlation between basically quantumness here and performance. Okay, and I think my time is up, which is fine. So here are my conclusions. So on the one hand, I said that photons guided by axons could potentially enable classical as well as quantum communication in the brain. Then I talked about these radical pairs. We think that electron spins in these radical pairs might underlie a wide range of biological phenomena that are sensitive to magnetic fields, and the highlighted paper here in bold, that's our review. So if you are interested, maybe start with that one. And these results, none of this proves that entanglement is really important for the brain or for consciousness, but it's at least consistent with this idea that large-scale entanglement might be fundamental for consciousness. So thank you very much.
Thank you. Christoph, thank you very much. Anirban is going to come up and set up for the next. But I'd like to ask, are you still there, Christoph?
Yeah, I'm here.
Do you know the myelin around the axon? You show that the light seemed to be in the myelin, and it's actually one layer wrapped in a spiral around the axon. And I've heard suggestions that the photons might reverberate in there as a sort of short-term memory, like the sound of the ocean in a conch shell, from that sort of thing. Have you looked at that, or do you think that's possible?
I mean, I think they would go through there very fast. So, any kind of memory effect would be very, very short term, I think. So I think it would be more like an optical fiber, so it basically goes through there at the speed of light. So like very fast, which also could be an advantage, right? From the point of view of information processing. I think the memories would have to be the spins.
All right, thank you. We have to move on. That was a great talk. I'm sorry for no more questions, but thank you very much. Round of applause.